Welcome to our video on how to use the ESMFold model in Python. In this video, we will look at how to use ESMFold to go from a protein amino acid sequence to a predicted protein structure. ESMFold has been shown to provide highly accurate protein structure predictions while not requiring a multiple sequence alignment of the input sequence. This makes it both fast and easy to use. So let's dive into the model. All the code shown in this video is available in our GitHub repository, which I've linked in the video description. In the repository, you will also find an environment file listing all required dependencies, and this mainly includes PyTorch and the Hugging Face Transformers library. Once you've followed the instructions in the README file to set up the Conda environment, you can open and follow along with the provided Jupyter notebook. First, we load the model. The initial load may take some time since the model parameters need to be downloaded. However, subsequent runs will be much faster. Depending on whether you're using a CPU or a CUDA-enabled GPU, we move the model to the appropriate device. We then switch the model to evaluation mode, since we only want to use it for inference and not for training. To begin, I will demonstrate a small toy example with a protein amino acid sequence with only 10 amino acids. Providing the input is straightforward. We simply store the protein amino acid sequence as a string and pass it to the tokenizer that we've loaded above. If we have a look at all the inputs generated by the tokenizer, we can see that each amino acid in our sequence is mapped to a unique token ID. In addition, we obtain an attention mask, which is set true for all 10 positions in our input sequence. We can then simply pass these inputs through the model. After inference, the model outputs a pLDDT score for each residue. pLDDT stands for Predicted Local Distance Difference Test, and it provides an estimate of how confident the model is in the predicted structure at each amino acid position. We can compute the average pLDDT score across all residues to obtain a confidence estimate for the predicted structure. This value ranges from 0 to 100, where higher values indicate higher confidence. For our toy example, we obtain a value slightly above 70, which suggests that the model is reasonably confident in the predicted structure. Next, we convert the predicted structure into a PDB file. This can be done easily using the model.output-to-PDB function. We save the result as example.pdb. If we inspect this file, we see that for each amino acid in the sequence, all corresponding atoms are listed along with their three-dimensional coordinates. To simplify this whole process, I've written a small helper function. And this function performs all of these steps in a single call. This function takes a protein sequence and an output file name as an input and directly saves the resulting PDB structure. We can now apply this function to a real protein sequence. For this example, I use a PETase sequence with UniProt ID E9LVH9. PETase is an enzyme capable of degrading PET plastics into smaller molecules. When we run the model on this sequence, we obtain a high estimated accuracy of over 80, and the predicted structure is saved as a PDB file. Since many proteins are multimers, which means that they consist of multiple protein subunits, we will now briefly look at how to predict the structure of a protein complex composed of multiple sequences. The recommended approach is to concatenate the individual sequences using a linker composed of glycine amino acid residues. In this example, we use a linker of 25 glycine amino acids. We take the first sequence, append the linker, and then append the second sequence. When preparing the model inputs, we additionally apply a large positional offset to the second sequence, for example 512. This ensures that both sequences are treated as part of the same input while being positioned far apart in the sequence space. As a result, the model understands that they represent separate subunits rather than a single continuous chain. We then run the inputs through the model as before. After prediction, we remove all atoms corresponding to the linker residues and proceed with the remaining structure. The final predicted 3D structure is saved to a PDB file, and we can again compute the mean pLDDT score to estimate the model's confidence. If we apply this approach to two toy sequences, we obtain a resulting PDB file along with an estimate of how accurate the model believes the predicted complex structure is.